Hello everyone, and thank you for joining me today. In today's lesson, we will be exploring receptor tyrosine kinases, referred to as RTKs, and their role in cancer. RTKs are essential cell surface receptors that regulate how cells grow, divide, and respond to signals from their environments. When these receptors become dysregulated, they can contribute to the development of progression of many cancers. Throughout this lesson, we will first examine how RTKs normally function. Then we will discuss how changes in these signaling pathways promote cancer. And finally, we will explore how this knowledge has led to the development of targeted cancer therapies. Let's begin by taking a look at our lesson objectives for today. Before we dive into the lesson, I would like to briefly review our lesson objectives. By the end of this presentation, you should be able to describe the basic structure and function of receptor tyrosine kinases, referred to as RTKs, and explain how they become activated during normal cell signaling. We will then build on that foundation by examining how mutations and other alterations can disrupt the signaling pathways and contribute to cancer development. And finally, we will discuss how our understanding of RTK signaling has led to the development of targeted therapies that are now used to treat several types of cancers. By understanding both the normal biology and the disease process, you will see why receptor tyrosine kinases remain one of the most important therapeutic targets in oncology today. Now that we have reviewed our lesson objectives, let's begin by discussing what receptor tyrosine kinases, which you will hear me refer to as RTKs, are. So RTKs are transmembrane proteins which function as cell surface receptors. Their role is to receive signals from outside of the cell and relay that information to inside of the cell, allowing cells to respond appropriately to their environment. One of the first things that we will notice in this figure is that RTKs are not a single receptor. Instead, they make up a large superfamily consisting of approximately 20 different receptor families in humans. For example, this first family is the ERB family, which includes the epidermal growth factor receptor, EGFR, along with ERB2, ERB3, ERB4. EGFR is one of the best studied RTKs because abnormal EGFR signaling is involved in several types of cancer. The figure also shows other well-known RTK families, including the insulin receptor, which regulates glucose metabolism, platelet-derived growth factor receptor, which contributes to cell growth and tissue repair, and the vascular endothelial growth receptor, which plays an essential role in angiogenesis and the formation of new blood cells. Now, just because these receptor families bind to different ligands and perform different biological functions, it is important to notice they all share a similar intracellular tyrosine kinase domain. This conserved domain is the defining feature of the RTK and is responsible for initiating intracellular signaling after the receptor is activated. Because RTKs regulate critical cellular processes like growth, proliferation, differentiation, metabolism, and survival, their activity must be controlled very tightly. When that regulation is then disrupted, the signaling pathways can become overactive and contribute to diseases such as cancer. Now that we have identified the different receptor tyrosine kinase families, RTKs, and the structural feature that they all share, let's examine how their unique structures allow them to recognize different ligands and activate intracellular signaling. As we dive into the structural mechanics of RTK activation, we're going to begin with the extracellular ligand binding region that is going to be shown here in panels A, B, C, and D. Each receptor family has a unique extracellular structure that is designed to recognize a specific ligand. This is specifically important because it ensures that the cells respond only to that appropriate signaling molecule. I want you to take a look at here panel A as an example. This shows the TRK-A receptor binding to our nerve growth factor. In panel B, this illustrates the KIT receptor interacting with our stem cell factor. Panel C has the fibroblast growth factor, FGFR, binding to the fibroblast growth factor down here. And then in panel D, we have the ERB family, including EGFR, interacting with the epidermal growth factor shown here. Although these receptors recognize different ligands, they all accomplish the same objective. Their objective is to detect signals outside of the cell, initiate that communication within the cell's interior. Now moving our attention over here to panel E. Once a ligand has been recognized, that extracellular event must be transmitted across that plasma membrane, and panel E illustrates that different receptor families undergo slightly different structural changes within their intracellular kinase domains during that activation. Even though the activation mechanics vary, the end result is very much the same. The tyrosine kinase domain became enzymatically active. Now I want you to ask yourself, why is this important? Why is understanding this important? If your answer is because activation of the kinase domains prepares the receptor to phosphorylate tyrosine residuals, initiating the intracellular signaling events that regulate cell growth, survival, differentiation, and metabolism, you would in fact be correct. That is why this is important. Panel F here highlights an interesting example using the EGFR kinase domain. Unlike many enzymes that function independently, EGFR becomes active throughout the asymmetric kinase dimer. One kinase domain acts like the activator, while the other functions as the receiver. This interaction stabilizes the active conformation of the receptor and allows that signaling to proceed efficiently. Now, this is a lot. So the most important takeaway from this figure is that RTKs exhibit considerable structural diversity in how they recognize ligands and become activated. However, despite these differences, they do all share the common goal of activating an intracellular tyrosine kinase domain that initiates a downstream signaling. This conserved signaling function is one reason RTKs are so effective at regulating numerous cellular processes. Now that we have discussed how RTKs become activated, the next question to think about is what happens after activation? Once the intracellular tyrosine kinase domain is activated, it then begins recruiting signaling proteins, which transmit information throughout the cell. Rather than activating a single pathway, one receptor can initiate several signaling pathways at the same time, allowing the cell to coordinate multiple biological responses. In panel A here, let's look at RTK. Everything begins with the activated receptor. The phosphorylated tyrosine residuals on the receptor acts as a docking site for adapter proteins, which then assemble signaling complexes and relay the signal to downstream pathways. So let's take this pathway down to GRB2. The first major pathway is the MAPK-ERK pathway. Adapter proteins like GRB2 recruit additional signaling molecules, eventually activating the RAS and the MAPK signaling cascade. This pathway ultimately activates ERK, which then moves into the nucleus and regulates gene expression, resulting in the MAP-ERK pathway primarily controlling cell proliferation, differentiation, and progression throughout the cell cycle. The second major pathway is the PI3K pathway. Activation of the PI3K leads to the production of the PIP3, recruiting proteins like PDK and AKT to the plasma membrane. Once activated, AKT promotes cell survival, stimulates cellular growth, and regulates metabolism by preventing apoptosis and supporting protein synthesis. The third major pathway is a PLCY pathway, shown here in panel B. When understanding this pathway, it is important to remember that IP3 stimulates the release of calcium ions from intracellular stores, but DAG activates protein kinase C, known as PKC. These signaling molecules then regulate a variety of cellular processes, including the secretion of migrational cytoskeleton remodeling and gene expression. Although we've discussed these pathways separately, it is important to remember that they do not function independently. Cells integrate signals from multiple pathways simultaneously to coordinate that normal cell growth, survival, differentiation, and metabolism. When RTKs become dysregulated, this coordinated signaling is then disrupted, which leads to persistent activation of pathways which promote cancer development. Up to this point, we've discussed RTKs as they function in healthy cells. Under normal conditions, RTK signaling is carefully regulated. A receptor becomes active only when it is needed and once the signal has been delivered. The pathway is turned off, similar to a light switch. You can turn it on, then you turn it off. This balance is important to note because cells should only grow, divide, or survive when it is appropriate. However, cancer changes that balance. Instead of receiving occasional growth signals, cancer cells often experience continuous RTK activation, meaning that the receptor remains active much longer than it should. This persistent signaling then causes the cell to behave as though it is consistently receiving instructions to grow and survive. Now we're going to take a look at this figure over here. We're going to focus at the ERB-B receptors family, which is going to include the EGFR. Think back to your previous slides. The activation of a receptor then triggers several intracellular signaling pathways. And in cancer, that same process will still occur. However, the receptor will not stop signaling now. So looking at the ERB ligands, why does this happen? Well, one reason is because the tumor microenvironment supplies an abundance of growth factors or ligands. These ligands then continually stimulate the receptor, allowing the signaling to persist far beyond what would occur within a healthy cell. Now think about the why. Why is persistent signaling so dangerous? First, the PI3K, AKT, and mTOR pathway, shown here, promotes cell survival. Normally, if a cell becomes damaged beyond repair, it will undergo apoptosis, programmed cell death. This protects the body from accumulating abnormal cells. But when this pathway continuously is active, cancer cells receive survival signals, which then help them avoid cell death. Instead of dying, they continue to live and accumulate additional mutations. The second major pathway is the RAS, RAF, MEK, MPAC pathway. Earlier, we learned this pathway regulates normal cell proliferation. Imagine what would happen if that pathway never turned off. The light is always on. The cell is now receiving repeated instructions to progress through the cell cycle, resulting in an uncontrolled cell division, a hallmark of cancer. So the light switch is always on, your electric bill is sky high, but instead in cells, we now have our cancer. The PLC-Y pathway contributes a different way. It helps regulate intracellular signaling through that calcium release and activation of the PKC protein kinase C. However, these signals influence gene expression, cell movement, and the communication within the neighboring cells. However, in tumors, these changes can increase the cell's ability to invade surrounding tissues and eventually metastasize. Now, taking a step back, focus on this figure as a whole. Instead of focusing on every individual protein, I want you to notice how one activated receptor connects to multiple signaling pathways. This illustrates a very important principle in cancer biology, which is one abnormal receptor can disrupt many different cellular processes simultaneously. That is why RTKs are such an important therapeutic target. If we can block the abnormal receptor, we do not just interrupt one pathway. We reduce signaling through many pathways that the tumor depends on for growth and survival. This now brings us to our next question. Are these receptors equally important in every type of cancer? So, our previous question, are the receptors that we saw on the prior slide equally important in every type of cancer? Well, the answer would be no. One of the most important concepts in precision medicine is that different tumors have different molecular characteristics. Even if two patients are diagnosed with the same cancer, the proteins driving tumor growth may not be the same in each patient. So here we have three graphs. This figure will compare the average expression of ERB receptors and several of their ligands in three common cancers. Our first one being breast carcinoma, second being non-small cell lung cancer, referred to as NSCLC, and colon carcinoma. Instead of focusing on the exact percentages, I want you to look at the overall patterns. We're going to focus on our breast carcinoma graph first. Notice that several members of the ERB family and their ligands are expressed at a relatively high level. This tells us that multiple signaling molecules may contribute to tumor growth in breast cancer, which helps explain why therapies targeting the ERB family have become an important part of treatment for certain patients. Now, compare that to our non-small cell lung cancer. Here we see a different expression profile. EGFR expression remains high, but the relative expression of other receptors and ligand changes. This reminds us that tumors arising in different tissues often depend on the signaling mechanisms even when they involve the same receptor family. Finally, look at our colon carcinoma graph. Once again, the expression pattern is now changing. Some receptors and ligands remain highly expressed, while others are present at a much lower level. These differences influence how tumors respond to the targeted therapies and help explain why a treatment that is effective for one cancer may not directly produce the same results in another. The takeaway for these three figures is for you to see that cancer is not a single disease. Even when tumors involve the same receptor family, the pattern of the receptor and the ligand expression can vary considerably depending on the tissue of origin. Due to this variability, modern oncology increasingly relies on molecular profiling to identify which receptors are driving an individual's patient's tumor before selecting a targeted therapy. Now, before we move on the previous slide, we saw different cancers expressing different levels of the ERB receptors, which raises an important question for us. Why do some tumors produce so much receptor activity in the first place? Think about it. The answer typically lies in the genetic alterations. Cancer cells frequently acquire changes in their DNA that either increase receptor expression or alter the receptor itself, allowing signaling to occur more frequently or even continuously more than it should. Let's start with the EGFR, one of the most extensively studied RTKs in cancer. Notice that there are several different types of genetic alterations listed. Although the specific mutations differ, they generally all fall into a few major categories. Gene amplification. The first is this one, gene amplification. Normally, a cell contains 2 copies of the EGFR gene, but in some tumors, extra copies are produced. More copies of the same gene lead to a greater receptor production, increasing the cell's sensitivity to growth signals and promoting excessive signaling. This table also shows a list of truncations, deletions, and tandem duplications. Rather than increasing the numbers of receptors, these alterations change the receptor's structure. Structural changes can affect how the receptor functions, sometimes allowing it to remain active when it normally wouldn't. In other words, the receptor begins to send growth signals even in the absence of a normal regulation. Another important category to understand is the point mutations within the kinase domain. Remember from earlier in the lesson, we talked about kinase domain functions as a signaling engine of the receptor. Well, mutations within this region can increase kinase activity, causing persistent activation of downstream signaling pathways that promote the cell growth and survival. All right, now let's study the ERB2. Clinically known as HER2, similar to alterations do occur here, gene amplification, splice variants, mutations within the kinase domain. These changes can likewise enhance receptor signaling and contribute to tumor progression. The table itself is not one that you are needing to memorize. We're not going to memorize every single mutation or every single exon number, but instead you are to recognize that cancers have different ways to disrupt normal RTK regulation. They may produce more receptors, alter the structure of a receptor, or increase the kinase activity. Regardless of the mechanism, the result is the same: persistent activation of signaling pathways that promote uncontrolled proliferation and survival. Now, the most important question in our lecture today that still remains is, do all patients have the same likelihood of developing these mutations? Well, that's a really good question, but the answer is no. One of the major discoveries in cancer biology is that the EGFR mutations are not distributed equally across all patient populations. Certain clinical characteristics are associated with a much higher probability of harboring an EGFR mutation. This table is going to show the incidence of EGFR, tyrosine kinase domain mutations within patients in non-small cell lung cancer. Rather than memorizing every single percent, your focus should be understanding the trends, as the trends have important implications for patient care. So we're going to begin with the smoking history, smoking or former smokers, and then never smoked. We're going to be breaking it down by looking at the smoking history first. So patients who have never smoked have a substantially higher frequency of EGFR mutations than smokers or former smokers. This finding demonstrates that lung cancer is not just a single disease with a singular cause. Instead, tumors can develop through different molecular pathways, and those pathways can influence which genetic alterations are then present. Now, moving on, we're going to examine the patient's sex. We have male and we have female. EGFR mutations are identified more in the female patients. Although researchers continue to investigate the biological reasons for the difference, these observations help clinicians estimate the likelihood which a patient's tumor contains an actual EGFR mutation. Now moving on to the histologic type. Examining this type of tumor, patients with adenocarcinoma demonstrate a higher incidence of the EGFR mutations than patients with non-adenocarcinoma tumors. This is one of the reasons why molecular testing has become such a routine part of evaluating patients diagnosed with lung adenocarcinoma. Finally, let's acknowledge the differences based on ethnicity. In this study specifically, EGFR mutations were reported more frequently in East Asian population than in non-East Asian populations. Findings like these highlight the importance of studying diverse patient populations and also remind us that prevalence of specific genetic alterations can vary across demographic groups. The takeaway for you here is, as we went through this table, the cancer treatment is becoming increasingly personalized. We are no longer selecting therapies based on where a tumor is originating from. Physicians and pathologists now examine the tumor's molecular profile to determine whether it contains specific mutations that can be targeted with precision therapies. Now, before we move on, I want to leave you with this thought. Once an actionable mutation has been identified, how do we target those abnormal kinases? If your answer to my previous question was targeted therapy, ding, 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 you have answered correctly. Targeted therapies, specifically tyrosine kinase inhibitors, TKIs, is how we target the abnormal kinases driving the cancer. These medications are designed to selectively inhibit abnormal kinase activity, preventing cancer cells from receiving the continuous growth and survival skills, which promote tumor progression. Unlike traditional chemotherapy, which affects both healthy and rapidly dividing cells, TKIs are designed to target specific molecular abnormalities. This approach is one of the foundations precision medicine, where treatment decisions are based on the genetic characteristics of an individual's patient's tumor. This table here does focus on chronic myeloid leukemia, also referred to as CML, which is driven by the BCR-ABL tyrosine kinase rather than a receptor tyrosine kinase. However, the important principle applies across all oncology. Once scientists identify the abnormal kinase responsible for a driving cancer, they are able to develop therapies which specifically inhibit that protein. That same precision medicine approach is used in cancers driven by the EGFR and other RTKs. Now let's look at the treatment column. This table is going to summarize several major clinical trials evaluating different TKIs, including imatinib, dasatinib, nilotinib, and bosutinib. Let's not focus on the individual trial names, but instead the patterns that these studies revealed. Each successful generation of TKIs were developed to improve treatment responses, achieve deeper molecular remission, and provide longer lasting disease control. The first two outcomes, CCYR and MMR, are measured are going to be our complete cytogenic response, our CCYR, and our major molecule response, our MMR. These measurements will then indicate how effectively treatment suppresses the leukemic cells responsible for the disease progression. As the response rates improve, clinicians then gain confidence to which abnormal kinase activity is successfully being inhibited. Now, let's look at the outcomes most meaningful for our patients. Event-free or progression-free survival, or EFS-PFS, which measures how long patients remain free from disease progression, while overall survival reflects a percentage of patients remaining alive during long-term follow-up. Across these clinical trials, survival rates remain consistently high, proving the remarkable impact that TKIs have had on patients. Our overall takeaway from examining and breaking down this table is that decades of research in cell signaling have translated directly into better clinical outcomes. Earlier in this lesson, we have discussed receptor structure, kinase activation, signaling pathways, and genetic mutations. All of those discoveries have provided the scientific foundation for designing drugs which specifically interpret abnormal kinase signaling instead of just a broad attack on random, rapidly dividing cells. So, if these therapies are so effective, why do some patients eventually stop responding to treatment? Well, the answer to this question for you guys lies in one of the greatest challenges in cancer treatment: drug resistance. Although TKIs can produce remarkable clinical responses, cancer cells do continue to evolve over time. As they divide, they may acquire additional genetic changes, which reduce a drug's ability to effectively inhibit the abnormal kinase. When that happens, the cancer may begin growing again despite that ongoing treatment. Let's think back to when we first discussed mutations. Those mutations with the kinase domains can alter how proteins function. Well, those same molecular changes can also interfere with how well targeted therapies bind to the intended target. As a result, the signaling pathways that were once successfully inhibited may become active again, thus allowing the cancer cells to survive and continue proliferating. Again, this table does have a focus on CML, chronic myeloid leukemia, which is driven by the BCR-ABL tyrosine kinase, illustrating a challenge that extends across targeted cancer therapies. However, whether the abnormal protein is RTK, like EGFR, or a non-receptor tyrosine kinase, like BCR-ABL, resistance can develop through additional molecular changes, so it does not matter. Knowing and understanding these resistant mechanisms will then help develop newer and more effective therapies. Here we see several second and third generation TKIs. These drugs were developed after researchers recognized some patients no longer responded adequately to the first generation therapies, like imatinib. Rather than abandoning target therapy altogether, researchers designed new inhibitors capable of overcoming many of the resistant mechanisms observed during treatment. These response categories here, CHR, MCYR, CCYR, help clinicians evaluate how well patients respond after switching to a newer inhibitor. So while the percentages are going to differ among the drugs used, the important message here is that patients were able to achieve meaningful clinical responses even after resistance develops. Now here we have our progression-free survival and our overall survival rates, which are going to remain favorable across these therapies. This shows the continued advance in targeted drug development can restore disease control and improve long-term outcomes for many patients. As a whole, this table for you should demonstrate that cancer treatment is constantly evolving. As tumors acquire new mutations, researchers continue to develop more selective and more effective inhibitors to overcome those changes. This is an ongoing cycle of understanding resistance and designing better therapies, which is one of the defining principles of precision oncology. As we begin to conclude today's lesson, I would like to leave you with one final question. Where is RTK research headed next? Well, over the past 2 decades, our understanding of RTK biology has completely transformed our cancer treatment. Scientists and their research have moved beyond simply identifying tumors based on where they originate in the body and now focus on understanding the specific molecular alterations driving each patient's care. This shift had made precision oncology one of the fastest growing areas in cancer research. One important advancement is the routine use of molecular profiling. Again, instead of assuming all tumors are going to respond the same way, physicians and pathologists analyze a tumor's genetic profile to determine whether mutations like EGFR are present. Identifying these actionable mutations help physicians select therapies that are more likely to be effective for an individual patient's care. We have also learned cancer is consistently evolving. As discussed, tumors can acquire additional mutation, which reduce the effectiveness of treatment over time. And due to this, researchers continue to develop next-generation TKIs that are capable of overcoming many resistant mechanisms and extending disease control for patients. Another important direction is the continued growth of personalized medicine. Rather than selecting treatment based solely on the type of cancer, clinicians are increasingly combining molecular testing, patient characteristics, and tumor genetics to develop individualized treatment plans. This approach improves the likelihood that patients will receive therapies specifically matched to the biological features of their disease. Finally, researchers are working to design more selective kinase inhibitors that effectively target cancer cells when minimizing damage to healthy tissues. New therapies are also being investigated in combination with other treatment approaches like immunotherapy, with the end goal of improving long-term outcomes and reducing the development of resistance. Needless to say, what began as fundamental research in cell biology has now led to treatments that are improving both survival and quality of life for many patients. It is important for you to know that as our understanding of RTK signaling continues to expand, so will the ability to develop increasingly precise, personalized, and effective cancer therapies. Thank you for your attention today, and I will be happy to answer any questions.